Hi, my name is Madeline Hasty, and my presentation is on a protein called ricin. Ricin is a toxic protein that is found in castor bean plants. The picture on the right is what this plant looks like. This protein acts as a hydrolase, cleaving an adenine bond to inhibit ribosomal activity. This protein has been found to enter the cell through endocytosis, but the specific mechanism is unknown. Ricin's structure is interesting, as it has two chains that are responsible for different actions in the inhibitory process. The protein as a whole weighs about 60.81 kilodaltons, and the structure can be seen to the right. Since each chain has different functions, I thought it necessary to present their structures separately. Both chains have about the same number of amino acids, but it can be seen that the A chain has a high amount of alpha helices, while the B chain has a high amount of beta sheets. These two chains work together to allow ricin to inhibit protein synthesis. There's a lot of current research on this protein, and I've included two important recent articles here. The first article's main focus is the mechanism by which ricin toxin induces the production of inflammatory cytokines. Liu et al. Are doing this research in Changchun, China. The second article's main focus is the detection of ricin in food in the case of suspected bioterrorism. Due to its toxic abilities, ricin is typically used in bioterrorism to fight against enemies. These studies are helping to decrease the amount of bioterrorism that occurs with it by finding strategies to detect the ricin toxin in foods and other materials. This research is being done by George Oliveira and Jose Mauricio Schneidorf in Alfenas, Brazil. The biochemical role of the two chains is important to understand the entire mechanism by which ricin acts to inhibit protein synthesis. The B chain functions to bind to glycolipids on the cell's surface. This binding is what allows ricin to enter the cell. Once it has entered, the B chain breaks off from the A chain, and the A chain moves towards the ribosomes. The A chain then hydrolyzes a specific adenine ring on the 28S ribosomal subunit. The cleavage of this bond results in improper functioning of the ribosome, which leads to the inhibition of protein synthesis and cell death. Although there are no diseases or conditions associated with ricin, it can be used as poison and can be lethal depending on the dose. Luckily, ricin poison is not contagious. However, there is no antidote. If the dose is high enough, ricin will be lethal and there is no stopping it. Interestingly enough, ricin has been used for good and bad. Due to its inhibition of protein synthesis, it has been experimented with to kill cancer cells. This research is very important in finding a cure to cancer. However, like other cancer treatments, ricin does not only affect the cancer cells, but normal cells as well. Ricin has also been used in wars as a form of bioterrorism to kill enemies on the battlefield. Other than bioterrorism and cancer research, ricin has had a substantial amount of research done in other areas of science. In an experiment done by Idkavivi and colleagues, depurination was done to evaluate the amount of adenine in a given sample. In this specific experiment, if the amount of adenine present is high, it means that the ricin toxin was cleaving the ribosomal adenine. If it is low, however, it means that the ribosomal adenine is intact and the ricin toxin was not inhibiting protein synthesis like it normally does. VPG, a known inhibitor of toxins similar to ricin, was used in this experiment to see how well it inhibited ricin. Different doses of VPG were used by removing a number of amino acids from the N-terminus of the compound. VPG 71 meant 70 terminal amino acids were removed, while VPG 111 meant 110 terminal amino acids were removed. As seen in the figure, there are three groups on the x-axis per each group of different VPGs. Each group represents a different concentration of that VPG group. So it goes 50 nanomoles, 100 nanomoles, and 200 nanomoles for each group. As you can see, for every VPG group except for VPG 111, as the concentration of VPG increases, the concentration of adenine decreases. indicating that VPG does decrease RTA activity in a dose-dependent manner in vitro. The fact that VPG 111 did not inhibit ricin activity suggests that the 40 extra N-terminal amino acids taken away are essential in the inhibition of ricin. These results are important because they support the hypothesis that VPG inhibits ricin activity, at least in vitro. Since ricin is poisonous and can be lethal, it is important to find a vaccine or antidote to prevent poisoning. Since it has been found that VPG inhibits ricin, future studies could be done to test if it would be a good antidote in vivo. A future direction that can be taken is creating a clinical trial using VPG in vivo to test if it would be a good antidote for ricin poisoning. Although ricin can be lethal, in small doses it resembles a case of food poisoning and will not kill an individual. By injecting small doses into multiple mice, it will not be lethal and will allow us to study if VPG inhibits ricin activity in vivo. The control, with no VPG injection, would be considerably safe since the dose would not be high enough to kill the mice and would give us an idea of the sickness that the mice would experience. Then, the experimental groups could contain different concentrations of VPG to help us determine what concentration of VPG works to inhibit ricin and if it actually prevents poisoning in vivo. If it is seen that VPG does inhibit ricin in vivo and prevents poisoning, further studies can be done to see if it will inhibit ricin poisoning when a lethal amount of ricin is injected. Thank you for listening to my presentation. Here are my references.